In this video, what we're going to be covering is the action potential. So in the first chapter, we went through some historical perspectives and we started talking about a brief introduction of neuroscience. In the second chapter, we started looking at neurons and glia. Last chapter, we looked at the membrane potential at rest. And now today, we're going to look at what's happening when we actually have a reversal of that membrane potential in the case of the action potential. This is where information is going to be integrated and sent down the axon to other cells. Now, just a little reminder, after this chapter, there will be an exam. So the first exam will be covering chapters 1 through 4. So it will be the historical perspectives, cells including neurons and glia, the different types, etc. The resting membrane potential, so the Nernst equation, Goldman equation, and then all the material that we go through in this chapter. The exams are not cumulative, meaning that each exam is going to be strictly based on just the material that we've gone over before it. So your next exam will be exam 2, which will cover chapters 5 through 7. And so you won't necessarily revisit questions directly related to these chapters, but these chapters are a solid framework for you understanding chapters in the future. Okay. So just a brief review from last time, the membrane potential, abbreviated V sub m, is the voltage difference across the membrane at a given moment. So when we look at negative 70 millivolts or negative 65 millivolts, that's the resting membrane potential. So that is the voltage difference across the membrane that exists when the neuron is not engaging in action potentials or sending information. An electrical potential is the relative voltage difference between two different areas. So this is just a more general term for a voltage difference. The resting membrane potential is the voltage difference across the cell membrane when the cell is at rest. So again, when the cell is not firing action potentials or firing and sending information, at rest, the membrane potential is going to be roughly negative 70 millivolts or negative 65. Again, I'm not going to ask you to differentiate between negative 70 and negative 65. Just know that negative 70 or negative 65 is roughly the membrane potential at rest. The equilibrium potential, abbreviated E sub ion or E sub Na plus. So if we're looking at the equilibrium potential for sodium, you would see it written as E sub Na plus or E sub Na. The voltage difference across the membrane that will balance out the force of diffusion for a given ion. So this is the voltage difference at which the flow of some specific ion, whichever ion you're looking at, across the membrane is equal to 0. So this is going to be dependent on the concentration of that ion on the inside versus the outside. For example, potassium's equilibrium potential is roughly about negative 80 or so millivolts, whereas the equilibrium potential for sodium is about positive 62. So that is due to the difference of concentrations inside versus outside of those respective ions. So the action potential is an electrical signal that is used to convey information over distances in the nervous system. What the action potential basically is it's a rapid reversal of the membrane potential in a neuron, such that normally inside the neuron you're resting at about negative 70 millivolts, and you can see down here at rest you're roughly around negative 70 millivolts. The membrane will become positively charged for a brief instant. Now, this helps to convey information over long distances. So remember when we were talking about those projection neurons, and we talked about our neuron, our cell right here, having a neurite known as an axon, which is able to send information down to a presynaptic terminal. So this is sending information away from the cell body in an anterograde fashion. Now, the neural code in action potentials will actually come from frequency and patterns. So the signal, the action potential, is a non-decremental signal where its strength is fixed. However, the thing that encodes intensity of a stimulus is going to be the amount of action potentials in a given segment and the different patterns that can arise, either in a burst frequency or in a sustained frequency, etc. So action potentials are also called spikes, discharges, or nerve impulses, and you can see those different terms used in the literature. But this is briefly what an action potential is. It's a brief change or reversal in the resting membrane potential so that the inside of the cell becomes positively charged, and then there will be a repolarization that occurs. So at rest, you're about negative 70 millivolts. Some stimulus is applied, whether that be opening channels through neurotransmitter activation, if actually applying a stimulus through like an electrode. This will change the membrane potential, make it more positive, and then there will be an opening of sodium channels, which leads to a rise in voltage. Eventually, the sodium channels will close, potassium channels will open, you will repolarize, actually go through an overshoot, and then and through the action of the sodium-potassium pump, you'll be able to return to rest. And don't worry, we're going to be going through this in a lot of painstaking detail. So the ups and downs of an action potential can be looked at through an oscilloscope. An oscilloscope is a device used to measure and study action potentials. This helps to record rapid changes in voltage. So it's similar to a voltmeter in looking at being able to read what the membrane potential is at any given moment, but what an oscilloscope does is it looks at a rapid change in voltage. So across this 1 to 2 milliseconds, we're able to see a rapid reversal in this membrane potential from negative to positive and then back down. So in the intracellular recording, you have a microelectrode right here that is placed in the inside in the cytoplasm. This is filled with concentrated salt solutions, and another electrode that's found in the extracellular space. Now, the extracellular recording, the electrode is near the membrane and another electrode. So what can happen is you'll actually see that these are roughly opposite of each other if you're measuring right outside. And the reason for that is because when we actually have this repolarization occur, you're actually going to have a release of ions as you're reestablishing that membrane potential. So here are the properties of the action potential, and all of this lasts for about 2 milliseconds. So these are different phases that you're going to want to know, particularly for the exam as well as for understanding this material. Negative 70 millivolts is our resting membrane potential. Negative 55 millivolts, roughly, so about negative 55 millivolts, is known as our activation threshold. This is the point at which an action potential will occur no matter what in an all or none response. The segment of the action potential which goes from negative all the way up to the top in the positive range is known as the rising phase or depolarization of the membrane. The inside of the neuron is positively charged with respect to the outside in the period known as the overshoot. Here we're at about roughly positive 40 millivolts. Now the falling phase is due to potassium actually exiting the cell. This is a rapid repolarization until the inside becomes more negative than our resting potential. So we actually have an undershoot. This is when the membrane potential is actually more negative than negative 70 millivolts. And then after hyperpolarization is the period where there's a gradual restoration of this resting potential from about negative 80 back up to about negative 70 millivolts. So terms that you should be familiar with when you're looking at the action potential. Depolarization. A depolarization is a change in the membrane potential from normal resting value to a less negative value. And whether you interpret that as less negative or more positive, it's up to you. Just usually, if you're already in the negatives, it makes more sense to think about it as a less negative value. Hyperpolarization, you have a change in the membrane potential from some normal resting value to a more negative value or less positive value. More negative makes a little bit more sense. So in our action potential diagram, this is a depolarization because you are becoming more positive or less negative. And the following phase is a hyperpolarization because you are becoming more negative or less positive. Threshold is the critical level of depolarization that must be achieved in order to trigger an action potential. And the activation threshold is roughly negative 55 millivolts. So at negative 55 millivolts, with a depolarization occurring that takes your neuron from negative 70 millivolts to negative 55 millivolts, once negative 55 millivolts is reached, you will fire an action potential. So depolarization beyond this threshold is going to always cause an action potential to occur in an all or none response. So the action potential is caused by depolarization beyond this threshold. And this is the activation threshold, negative 55 millivolts. This can be done in numerous ways. So this can be done through channels sensitive to membrane stretch, channels sensitive to neurotransmitters such as ligand-gated channels, or directly injecting electrical current through a microelectrode into the neuron. Now this is an all or none response. So if you think about a disposable camera, a Kodak disposable camera, if you've ever taken a picture with one, what happens when you push the button? The picture is taken. Now, nothing happens until you fully push down the button, right? Now, what happens when you fully push down the button? You can't change your mind halfway through that, right? As the camera flashes, you can't change your mind and say, okay, I don't want to take the picture anymore. It is an all or none response in the same fashion as a neuron firing an action potential. Once negative 55 millivolts is reached, an action potential will occur. Just like in a Kodak camera, once you hit the button, a picture will occur. And so action potentials can be seen in chain reactions such as puncturing your foot. Stretch membranes of nerve fibers are activated, opening sodium permeable channels, leading to an influx or an increasing amount of sodium coming into the cell. This will depolarize the membrane. If it reaches threshold, it will fire an action potential. Now, if you do not reach negative 55 millivolts, then you will not fire an action potential. So, the firing frequency of action potentials, and this just simply means the number of action potentials in a given segment of time, this helps to reflect the magnitude of the depolarizing stimulus or the depolarizing current. And this is one way that stimulation intensity is encoded. So if you think about intensity, if someone flicks you, so someone just lightly flicks you, versus if someone punches you, the action potential is always the same magnitude. However, what is able to be encoded is the intensity of the stimulus. If you think about being flicked versus being punched is a more intense stimulus, right? So the action potentials that carry that information to the brain are going to be more frequent. The action potentials aren't larger, they aren't stronger in and of themselves. The number of action potentials going to the brain will be different if you're punched versus if you're flicked. So a larger depolarization does not mean a larger action potential. The action potential will always be the same magnitude once the threshold is reached. So if you get to negative 55 millivolts, or if you get to some depolarization which takes you to negative 50 millivolts, both of these action potentials are going to be the exact same strength. The only thing that will differ is the amount of action potentials in a given segment of time. So firing frequency reflects the magnitude of the depolarizing current or the depolarizing stimulus. So the stimulus that led to the depolarization or the amount of current that led to a depolarization. Now, there is a limit to the rate at which a neuron can generate action potentials, and the max firing frequency is about 1,000 hertz. That's about 1,000 times a second. And the reason why that's the firing frequency is because at its highest level, an action potential takes about 1 millisecond. Well, 1,000 milliseconds is equal to a second. So you can't physically go faster than that. Now that's the max firing frequency. Now, there are two other terms that we should be familiar with when we're looking at the action potential. That's the absolute refractory period and the relative refractory period. The absolute refractory period is once an action potential is initiated, it is impossible to initiate another one for about a millisecond. The absolute refractory period is due to the fact that voltage-gated sodium channels are tightly closed. You cannot open those again for about a millisecond. The relative refractory period is the period where it is relatively difficult to initiate another action potential, but it can be possible. During the period of the relative refractory period, the amount of current required to depolarize the neuron is elevated above normal. So you just need more current or more stimulation to depolarize the neuron to activation threshold. So here are the different refractory periods. So here's our action potential right here, where we're at resting membrane potential. We're depolarized the cell, going up here to about positive 40 millivolts. During this period right here, where your neuron is being repolarized back down to a resting state, which is negative 70 millivolts, you're in the period of the absolute refractory period. Meaning, during this moment right here, you cannot fire another action potential. Now, during the phase where you are actually below negative 70 millivolts, you just need a higher amount of, or more current, or a greater stimulation, or a greater stimulus, in order to get you to negative 55. So if we change the pen color really quick, and let's change it to yellow. Here's, let's say, negative 55 millivolts, right? So this is negative 55 millivolts, here's negative 70. You need roughly negative 15 millivolts, or 15 millivolts of difference to get from negative 70 to negative 55, right? If you're down here at roughly negative 80 millivolts, in order to get you from negative 80 to negative 55, you need 25 versus 15. So do you see what I'm saying? In the relative refractory period, it's possible to fire another action potential, but you just need a greater stimulus than you normally would if you were at resting state. So here, we can look at injected current into a neuron and the ability of the neuron to fire an action potential. And each of these spikes is an action potential. So if the injected current here, during this first segment, does not depolarize the membrane, again, to its threshold, no action potential is generated. So there's no spikes during this period because the injected current was not enough for the neuron to reach negative 55 millivolts. So this graded potential decays back to resting state. Now, if we inject a little bit more current and sustain that injection, if it depolarizes the membrane beyond its threshold, action potentials will be generated. So, generation of action potentials. Now, if we take away that current, it goes back down to resting state. Now, the action potential firing rate increases as the current increases. So notice how we went from this level of injected current, now we're injecting even more. Notice how there's not some magical, there's not some magical change in strength of the action potential because we're injecting more current, right? We've injected more current, but the action potential's peak is still the same. However, in a given segment, we're firing more action potentials. So notice here, we're firing 1, 2, 3, 4, 5. In this segment, we're firing 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 action potentials. So what is encoding the intensity of the stimulus, or the intensity or the amount of injected current that we are using to depolarize the neuron? Firing frequency is what is encoding that information. So a really cool property of the action potential is that one thing that we can do is we can actually introduce foreign genes into neurons where we can actually shine light on them, which can allow for these channels to open or close. And we can control a cell's depolarization or hyperpolarization through the introduction of light. So channel rhodopsin is a photopigment which, when introduced into mammalian cells, will encode a light-sensitive cation channel, so cation being positively charged ions, permeable to sodium and calcium. So this opens rapidly in response to blue light, and an inward flow of cations is sufficient to produce depolarization beyond the activation threshold. Opposite of channel rhodopsin is halorhodopsin, which is a protein derived from single-cell microbes that will lead to an inhibition of neurons in response to yellow light. And this is due to the fact that there's an opening in a chloride pump. So in a mouse brain, neurons firing is controlled by light delivered through an optic fiber. So here's a mouse, through optogenetics, has had channel rhodopsin inputted into a region, and with stimulation through blue light, this will lead to sodium flowing in. Now when sodium flows in, this is a positively charged ion. If you're starting at negative 70 millivolts, and you're adding positively charged ions, this is going to make this value more positive. So let's say negative 66, negative 65, etc. So you see there's a depolarization here when the light is on. In the case of halorodopsin, this opens up a chloride channel. And if you're starting at negative 70 millivolts and you're introducing more negatively charged ions, this is going to cause the membrane potential to go down or become more negative or less positive. So negative 75, negative 80. And note, when you hyperpolarize a neuron, you're making it harder to fire an action potential, right? Because if right here is your activation threshold, you're getting further and further away from this point. So optogenetics is a really cool, innovative technique to be able to introduce and study the effects of depolarization, hyperpolarization on cellular circuits. So during an action potential, we go through phases of depolarization, where we have an influx of positive ions, sodium ions, and a period of repolarization, where we have an efflux or an exiting of potassium ions. So the current of a given ion is the net movement of that ion across membranes. So in this case, we're looking at the current of potassium. So if we're wanting to look at I sub K, this is potassium current, the movement of potassium ions across the membrane. This is going to be equal to the conductance of potassium multiplied by the membrane potential minus the equilibrium potential. So conductance is G. This is proportional to electrical conductance, and this is going to be the presence of potassium channels. Now remember, if you don't have potassium channels, or if you don't have channels in general, then no current can flow, because there's no way that the ion can go anywhere. So the membrane potassium current will only flow as long as the membrane potential doesn't equal the equilibrium potential. Remember that at the equilibrium potential, you have a cessation of movement, right? You have the electrical gradient that is going to be balanced with the concentration gradient. So the driving force on potassium is going to be the difference between the real membrane potential and the equilibrium potential. So if the membrane potential minus the equilibrium potential for a given ion is greater than 0, positive ions are going to flow out. This results in an outward current. If the membrane potential minus the equilibrium potential is less than 0, positive ions will flow in, and this is an inward current. So what I would like you to do is just take out a piece of paper or just sort of draw on something, because I want to be able to show you how this makes sense. So if the membrane potential minus the equilibrium potential is greater than 0, positive ions flow out. So in the case of potassium, let's say the resting membrane potential, or the membrane potential for a given neuron is negative 65 millivolts, right? Now, we subtract the equilibrium potential for potassium, which is roughly minus 80 millivolts. So our equation is negative 65 minus negative 80 turns into positive, and that will be 15 millivolts. 15 is greater than 0. This results in positive ions flowing out. And that's what we see. Now, in the case of sodium, let's say negative 65 millivolts is our VM, or our resting membrane potential, or our membrane potential here. Subtract that from the equilibrium potential of sodium, and that is, let's say, roughly 60 millivolts, let's say. Negative 65 minus 60 millivolts is going to be equal to negative 125 millivolts. That is indeed less than 0. Positive ions flow in through an inward current, and that's what we see in the real world looking at potassium and sodium ions and their current flows. So here's an ideal neuron, and this is looking at the action potential in theory. So we have our equilibrium potential for potassium at negative 80 millivolts, our equilibrium potential for sodium being equal to 62 millivolts. Now, in this case, there are no channels present. So we're assuming that all channels are closed, and the membrane potential is equal to 0. So if your conductance of potassium is 0, then there's no current flow, because anything multiplied by 0 is 0. Okay. Now, let's say we open up some potassium channels. The conductance of potassium is now greater than 0, right? So potassium channels are open, potassium is going to flow out. So the current of potassium current flows as long as the conductance is greater than 0 and the membrane potential of that cell is not equal to the equilibrium potential of potassium. So assuming that the membrane potential is not negative 80 and that there are channels that are open, you are going to have potassium current flow. Now, at equilibrium, there is no net potassium current because your membrane potential is equal to your equilibrium potential. An equal number of potassium ions enter and leave at the same time. So note that even if your conductance is greater than 0, your channels are open here, if your membrane potential is equal to your equilibrium potential, this will result in your conductance multiplied by 0. That will give you 0. So your current will be 0 at equilibrium. So the different phases of the action potential, you have the rising phase, which is due to an inward sodium current, and your falling phase, which is due to an outward potassium current. So when the membrane is depolarized to threshold, so remember, at activation threshold, you are going to have a depolarization occur. Your conductance of sodium increases, and this is what allows sodium entry. And as you get more and more positive ions coming into the cell, you are depolarizing the cell, meaning that you are driving the membrane potential to a more positive value. So at negative 80 millivolts, which is the equilibrium potential of potassium, your membrane potential is equal to the equilibrium potential, and you don't have any kind of movement. Now, as you open up more sodium channels, which is what occurs when you reach activation threshold, the conductance of sodium is greater than that of potassium. So your membrane potential moves towards the equilibrium potential for sodium. Now, it doesn't reach the equilibrium potential for sodium. So your sodium channels will actually close, and this is due to the fact that they're voltage gated. Once you reach about positive 40 millivolts, these voltage gated sodium channels close. In the period of repolarization, which is taking you from a positive value back down to negatives, your conductance of potassium, and these are voltage gated potassium channels, these open, allowing for potassium to flow out. This is what helps to bring down the membrane potential back down to resting state. So during the falling phase, you have a conductance of potassium which increases, allowing potassium to leave faster. Now, this will be sort of a brief review of concentration gradients and diffusion. So if you want to take out like a piece of paper, it's sort of hard for me to draw using this mouse, but inside your neuron, as we talked about last time, you have a lot more potassium inside the neuron, and you have a lot more sodium and chloride on the outside of the neuron. During an action potential, what happens is after activation threshold, you have a lot of sodium flowing into the neuron, right? So sodium, being a positive ion, is attracted to the inside of the cell because the inside of the cell is negative 70 millivolts, roughly. During this period of depolarization right here, sodium is flowing in. Once these sodium channels close, note that the inside of the cell is positive. Well, you have a lot of potassium inside the cell, and potassium carries a positive charge. Potassium doesn't want to stay inside the cell because of electrostatic repulsion. So when you open up channels, voltage-gated potassium channels, this is what causes the repolarization to occur. Potassium wants to go down its concentration gradient, 1. It's higher on the inside versus the outside, but it also doesn't want to be in an area that is positive. So Hodgkin and Huxley were able to use voltage clamp techniques, looking at clamping the membrane potential at any chosen value, to actually be able to hypothesize and propose the existence of sodium gates in axonal membranes, accounting for changes in the conductance of sodium by depolarization and inactivations by positive membrane potentials. And this was really cool and really groundbreaking research, particularly in the 1950s, using squid axons, being able to understand and propose that it's due to the presence of these channels and these gates which are allowing for the conductance of sodium to cause depolarization and positive membrane potentials to lead to inactivation of these channels. Now, the voltage-gated sodium channel is a very important channel, and this has a lot to deal with why the active potential occurs in the first place. Now note, this is voltage-gated, meaning that in order for this channel to open, allowing for sodium to come in, it is going to be dependent on the membrane potential. At resting state, you don't have these voltage-gated sodium channels opening. Once activation threshold is reached at negative 55 millivolts, these voltage-gated sodium channels do open. And we'll talk about why that occurs, because there's actually voltage sensors present in these proteins. So the structure of the voltage-gated sodium channel is it has transmembrane domains, simply meaning that there are segments of the protein that will go across the membrane. So here is the membrane. And you have transmembrane, which means that you have segments that are going to go across the membrane. There are four domains, 1 through 4, which are going to be composed of the entire protein. So note here, each of these domains, 1, 2, 3, and 4, these are subunits, and altogether, these are the tertiary structures of the protein, and then you have these coming together to form the quaternary structure. So each domain consists of 6 transmembrane alpha helices, S1 through S6. So here is one domain, and then you have S1, S2, S3, S4, S5, and S6. For this class, S4 and S6 are going to be very important to understand. Now, voltage-gated sodium channels open and close due to changes in membrane voltage. So when the membrane is depolarized, meaning that is a more positive value, the molecule twists into a configuration that allows for sodium passage to occur through the pore. So when the membrane is depolarized, this actual channel will change and twist into a different configuration, allowing for the channel to open, causing sodium to flow in. At resting membrane potential, these channels are in such a configuration whereby the channel does not open, allowing for sodium to go in. This is what makes it really great. This is why there's an actual resting membrane potential. You're not allowing for sodium to flow in. So the structure of this voltage-gated sodium channel allows for gating and pore selectivity. The pore loops are assembled into a selectivity filter, meaning that you're not just going to allow any kind of cation to flow through. The voltage-gated sodium channel is about 12 times more permeable to sodium than it is to potassium. And in this, sodium ions are stripped of most of their associated water molecules, so these shells of hydration, as they pass through the channel. So notice here at resting membrane potential of roughly negative 65 millivolts versus negative 40 millivolts, there's a change in configuration whereby this voltage sensor seen in the S4 transmembrane section of a domain is actually changed in configuration when there's a depolarization occurring. So the voltage sensor resides in segment of S4 of the molecule. Positively charged amino acid residues are regularly spaced along the coils of the helix. So let's change this pin color to green. So right here along the coils of the helix, you'll see that there are positively charged amino acid residues. Now this is important for why this acts as a voltage sensor. So if you change the membrane potential, you move this segment. So you move this S4 segment. And why is that? Well, if you're depolarizing the cell, you're making the cell more positive. And due to electrostatic repulsion, these positively charged sections are going to move. Now when you move this S4 segment, you're changing the configuration of this protein. allowing for this channel to open up, allowing for sodium to flow in. So depolarization of the cell membrane will twist this S4 segment, and this conformational change is what causes the gate to open, allowing for sodium to flow in. Now, the reverse is also true. So when you think about the repolarization segment, what happens when you reach about positive 40 millivolts? Around positive 40 millivolts, now you're in a positive area, so you actually have a change in the configuration whereby you have this reestablishment of this normal native structure. So you have a closing of the channel occurring. This will prevent sodium from flowing in anymore. And this is important because if you didn't have these voltage-gated sodium channels, then you would have no ability for your cell to repolarize or be able to go back down to resting state, or to be able to fire action potentials in that matter. So patch clamp methods are important techniques used to study ionic currents passing through individual ion channels. And this is what electrophysiologists are able to do in studying each of these individual receptors or pores or channels. So what's done in patch clamp methods is you make a tight seal between the membrane and the tip of the electrode, leaving ions only one path to take. So using a gigaohm seal, what you'll do is through this pipette tip, be able to just make a seal between the membrane and the tip of the electrode. So you have this single sodium channel, or whatever channel you want using patch clamp methods, but you can tear away this membrane patch, and you can measure ionic currents as steady voltages are applied across this membrane. So you can inject current or voltage changes across this single patch of membrane and be able to measure whether or not these channels open or close. So voltage-gated sodium channels open with very little delay. They stay open for about 1 millisecond and then they close, and this is the period of inactivation. This is why you have an absolute refractory period. Why there is a period of time where you cannot fire an action potential. And the reason for that is because these sodium channels are completely closed. They can't be reopened again immediately by depolarization until the membrane potential returns to a negative value near the threshold, and that's that after shoot. So the absolute refractory period is where the voltage-gated sodium channels are inactivated. This is the period of time whereby it is impossible to initiate another action potential. So here's your voltage-gated sodium channel. Sodium will flow in when the membrane is depolarized. During the absolute refractory period, your channel is completely closed. And then once you get back into the relative refractory period or the resting membrane potential, your sodium channel is ready to go again. So in certain genetic diseases known as channelopathies, these can be caused by alterations in structures and functions of ion channels impeding the current of either sodium or potassium. So generalized epilepsy with febrile seizures is due to slowing and activation of the sodium channels, which essentially works to prolong action potential, so too much electrical activity. In experimental settings, toxins can be used as tools to be able to measure specific sites on channels and as well as specific activities in channels. So the pufferfish toxin, tetrodotoxin, clogs sodium permeable pores, and it actually binds to specific sites on the outside of the channel, causing sodium channels not to work. And if you don't have sodium channels working, then you can't fire action potentials. If you don't fire action potentials, then you can't have information processing going on. Therefore, you die. That's why sushi chefs have to go through extensive amounts of training to be able to prepare fugu or pufferfish. Because if you don't prepare it correctly, then the tetrodotoxin is able to enter into the sushi eater's body and can kill them. One of the papers that I have chosen for the reading assignment is a really good paper on TTX as like a pain relief agent. And I think it's pretty interesting because you can use TTX to be able to study different channel activities. And you can see how this can be applied in a good experimental setting. And then red tide saxitoxin is a sodium channel blocking toxin. So a variety of toxins can affect channels. So batrachotoxin or batrachotoxin from frogs can block inactivation, so channels remain open. So this will block the ability of the channel to be inactivated, so you have a continual opening of the sodium channel. Similar effects are seen from veratridine and aconitine. Sodium channel inactivation is also disrupted by toxins from scorpions and sea anemones. So a lot of these toxins from nature work directly at the level of channel pores. So differential toxin binding sites provide clues about 3D structures of sodium channels. So if you're able to look at where these toxins bind and how these toxins interact at the level of the channel, then you can be able to sort of get an idea of the way that the sodium channel, potassium channel, or whatever channel you're looking at, how it is arranged in 3D space, and where the toxin's binding, and you can hypothesize about what's going on at that particular level. So you can also study consequences of blocking action potentials, so blocking impulses in nerves or muscles. And so a lot of poisons, like nerve agents, will actually work at particular synapses or particular receptors, and this is what causes, ultimately, death. And we'll talk about that when we start talking about neurotransmitter systems. Now, just like voltage-gated sodium channels, you also have voltage-gated potassium channels. Now, voltage-gated potassium channels and sodium channels both open in response to depolarization. They're voltage-gated. However, potassium gates open much later than sodium gates. These voltage-gated potassium channels will actually open when the membrane potential is positive. These are known as delayed rectifier channels, where the conductance of potassium serves to rectify or reset the membrane potential. So remember, if you have your action potential looking like this, let's change the pen color to green, your voltage-gated potassium channels will open at this point, allowing for the membrane potential to rectify or reset. You have an outward flow of potassium going in this repolarization period. So there are many different types of voltage-gated potassium channels which help to function to diminish any further depolarization. This gives potassium a path to leave the cell across the membrane. Now, if you're at a positive value, right, so positive 40 millivolts, and you're allowing for potassium, which is a positive ion, to leave, then you're getting rid of more and more positive ions. Thus, your membrane potential is going to become more negative or less positive. So the structure of the voltage-gated potassium channel, there are 4 separate polypeptide subunits that join together to form a pore at which potassium is able to flow through. Now, these proteins are sensitive to changes in the electrical field across the membrane, in the same idea that voltage-gated sodium channels were. Now, when the membrane is depolarized, the subunits are believed to twist into a shape that will then allow potassium to pass through. So key properties of the action potential that we've already talked about and that we're just going to go over again is the threshold period. So this is roughly negative 55 millivolts, the moment at which these voltage-gated sodium channels will open up, allowing for sodium influx to occur. This period of sodium influx is known as the rising phase of the action potential. The overshoot is the period at which you are approaching the equilibrium potential for sodium. The falling phase is due to potassium efflux. So voltage-gated sodium channels are closed, whereas voltage-gated potassium channels or delayed rectifier channels are open. These potassium channels stay open as long as the membrane is depolarized, which causes hyperpolarization to occur. Now, these are delayed channels, meaning that your voltage-gated channels actually close when you go beyond resting state. And thanks to the sodium-potassium pump, you actually get this reestablishment of your ions. So the absolute refractory period is due to the voltage-gated sodium channels becoming inactivated. And the relative refractory period is the period at which you have this undershoot here, where it's a little bit harder to go from this hyperpolarized state up to the period of threshold. It takes a larger current or a larger stimulus to be able to reach that period. However, once you reach negative 55 millivolts, your action potential will fire. Now, how are action potentials conducted and how are they propagated? So, action potentials are propagated in an orthodromic fashion, meaning that they travel in only one direction. Not the band, but they only travel in one direction, from the soma down to the terminal. So it goes down the axon to the axon terminal. So if you have a cell right here, and the axon, which goes down here to the terminal button, the action potential will travel from the soma or the axon hillock, which is the initial segment, down to the terminal. Only in experimental settings can action potentials travel in an antidromic fashion, or a backward propagation. Now, the reason why it only goes in one direction is due to the fact that periods that have just had the depolarization occur are now in an absolute refractory period. So due to the axonal membrane being excitable along its entire length, the action potential will propagate without decrement. It's a non-decremental signal. Meaning that the action potential is a particular strength here, as it is here, as it is here. It's the same strength. The typical conduction velocity for an action potential is about 10 meters per second, and the typical length is about 2 milliseconds. So it's like a match to a fuse, and I have a video right here that I'll show you.
This length of axon can be likened to a piece of fuse. The burning match under the axon represents an electrical event that generates an action potential. This event normally takes place at the axon hillock. Recall that to trigger an action potential, the electrical event must depolarize the axonal membrane beyond its threshold. That is, the membrane potential has to become positive enough to initiate the action potential, which is an all-or-nothing event. Once the membrane reaches that threshold, voltage-gated sodium channels open up, and sodium rushes in, as reflected in the upward deflection of the membrane potential. But how is the action potential propagated down the axon? Look at the area just ahead of where the action potential has just fired. It is becoming more depolarized. A millisecond later, the positive charge associated with the first action potential has spread to an adjacent region of the axon. This area reaches its threshold, and this segment of axon fires an action potential. The area of axon that fired an action potential a millisecond ago cannot become depolarized now. It is refractory because its sodium channels are inactivated. In this way, the action potential propagates in only one direction, like a fuse. Now, 2 milliseconds later, the positive charge spreads to the next adjacent region of the axon, and it reaches its threshold and fires an action potential. The region of axon that fired a millisecond ago cannot be depolarized. It is refractory. The area of the membrane that fired 2 milliseconds ago has now returned to its resting potential, but it is too far away to be influenced by the current action potential. In this way, the action potential migrates, like the fire on a fuse, toward the axon terminal, where it is delivered to another neuron. Now let's watch the whole process at high speed.
So in this fashion, you're going to have a traveling depolarization, because that's all the action potential is if you really think about it. It's a massive depolarization that's occurring, going down the length of the axon. So factors that influence conduction velocity of an action potential are the spread of it along the membrane. And this is dependent on axon structure. Now, here I have a little arrow going down here. If the axon is wide and there are few open membrane pores, then your action potential will travel faster. If the axon is narrow, with numerous open membrane pores, then your action potential will travel slower. In the general fashion, the larger the axon diameter, the faster your action potential will travel, as well as the number of voltage-gated channels that are physically there to excite the axonal membrane. So the speed at which the action potential is conducted will depend on how far the depolarization ahead of it will be able to spread. So thicker axons that have less open pores or channels are the quickest conductors of action potentials. So thick axons with membrane pores, the current will flow inside the neuron, where if you have thinner axons with membrane pores, the current will tend to flow across the membrane instead. So think about like a garden hose. So think about a garden hose that's very thin that might have a lot more holes. If the water flows down that garden hose, a lot of the water is going to leave through those holes, right? However, if you have a very large garden hose, with a very large diameter, with very few holes, you're going to get a lot more water that goes in a quicker fashion. So thin axons with more channels will conduct active potential slower. These tend to require greater depolarization to function, as more of the voltage-gated sodium channels must be open to ensure that the active potential does not fizzle out. So the voltage-gated sodium channels are how local anesthetics are able to work. Luckily, I hate the dentist, and I'm very glad that things like lidocaine and novocaine are working. So what happens with local anesthesia is that it binds to voltage-gated sodium channels, and this is in the S6 alpha helix of the fourth domain of this voltage-gated sodium channel right here. This is where lidocaine is able to physically bind. Now, smaller axons are affected before larger ones, because in smaller axons, more voltage-gated sodium channels must function in order for proper conduction of the active potential to occur down the axon. And we just talked about that in terms of the diameter of the axons and the presence or absence of more or less sodium channels. So how lidocaine and anesthetics actually get down to this binding site is through diffusion through the lipophilic nerve membrane, where it will actually bind and be able to inactivate the voltage-gated sodium channels. Now, if you've ever had anesthesia, what happens? You're unable to feel brief pain, right? So usually if you're going in for a dentist checkup, or if you need to get a root canal or anything like that, anesthesia is administered directly to the point where you're getting any kind of procedures done. By blocking voltage-gated sodium channels, you're preventing active potentials carrying pain information from reaching the brain. That's why during procedures such as in dentistry, you don't feel a lot of pain often, due to the fact that the local anesthesia is preventing those voltage-gated sodium channels from conducting active potentials. Other factors that influence conduction velocity and active potentials are the presence or absence of myelin sheaths. So myelin will help facilitate current flow. In a highly myelinated axon, this will increase the active potential conduction velocity. So a brief review from last time, you have Schwann cells, which are the myelin-producing cells in the peripheral nervous system, and oligodendrocytes, which are the myelin-producing cells in the central nervous system. Now this helps to facilitate current flow down the inside of the axon, which helps to increase active potential conduction velocity. And in the disease known as multiple sclerosis, this disorder is characterized by weakness, lack of coordination, and impaired vision and speech. And the reason why this is because in multiple sclerosis, you actually have myelin which becomes damaged, exposing the axonal membrane. And this causes the active potential conduction velocity to greatly decrease. So myelin sheaths or bundles of axons in the central nervous system and optic nerves are being attacked, which gives you these behavioral changes. So you have profound slowing of response time. So you can measure electrical responses at the scalp over optic nerves after stimulating the eyes, since saltatory conduction, which is this process, is disrupted. So in nerves affected by MS, you have a gradual eating away of this myelin, exposing this axon that lies underneath. So this is a process known as saltatory conduction, which occurs at nodes of Ranvier. And nodes of Ranvier are unmyelinated sections of axons that contain a large amount of voltage-gated sodium channels. This is what essentially allows the active potential to be reborn or regenerated along all the stretches of the axon. That's why at any given moment, your active potential is the same exact strength no matter where you are at along the axon. So in myelinated axons, active potentials skip from node to node, hence the term saltatory conduction. Now, there are cases of axons that are not myelinated, and those depend on what sections of the nervous system and which sensory systems you lie in. Some pain fibers are unmyelinated, whereas other pain fibers can be highly myelinated. Axons carrying information about touch will tend to be myelinated, joint position, etc. And that depends on the extent of how quickly you need those signals going to the brain. So here is a brief demonstration of saltatory conduction. And let's see if this opens up or not. Hoping that it does. Okay. Guess not. But this demonstration, see if you can go to this website. But this is briefly just showing why, in a case of saltatory conduction, why nerve signals or action potentials will travel quicker in a myelinated axon versus an unmyelinated axon. And the reason for that is because the myelin sheath essentially acts as an insulator where you don't have a loss of any of these, of any of this charge going down. So instead of having to go through the process of depolarization and hyperpolarization along the entire stretch of the membrane, you only have to do it at each node. So dendrites and cell bodies, or somas, don't generate action potentials because they have very few voltage-gated sodium channels. So by this time, you should understand that voltage-gated sodium channels are very, very important for the propagation and for the creation of action potentials. Now, the axon hillock is known as the spike initiation zone. This is the part of the neuron where an axon originates from the soma, and the sensory nerve endings are sites of depolarizations in sensory neurons. So here's the spike initiation zone, or the axon hillock, where you have a very high density of voltage-gated sodium channels. This is where the action potential will begin, and then you'll have a traveling of that information down the axon towards the terminal button down here. The spike initiation zone in sensory nerve endings will lie at the level of, let's say, your skin, will travel up through its process and will travel down and go into the central nervous system. So the differences in types and density of membrane ion channels can account for characteristic electrical properties of different neuronal types. So we've already discussed how neurons vary not only in shape, size, gene expression, and connections, but they can also have different electrical properties. So some neurons could fire action potentials relatively steadily. Others can show adaptation by firing rapidly at the beginning, but slowing down over time, and then others show burst patterns. So why is this the case? Well, if you've already thought about how ion channels are important for the creation of the action potential, this is the same reason why there are different electrical properties in different synapses, in different neurons, because there are different classes of channels which can exist. There are different properties of ion channels in the membranes of given neurons, and the different numbers of ion channels that may be present in given neurons. So each class of neurons has a particular signature based on the complex interactions, based on how itself is composed in terms of ion channels. So here are just some brief types of electrical behavior in neurons. We take a depolarizing injected current, and you see a steady frequency of action potentials firing. So as this injected current is depolarizing the membrane, you see an action potential, go back down, action potential, action potential, action potential, as long as this injected current is remaining. You could have adaptation, where they fire rapidly at the beginning and then slow down. So here, you have an injected depolarizing current, fires rapidly here, but then slowly adapts. Or you can have burst firing, where you have a lot of action potentials occurring in short segments, and then the neuron will show periods of adaptation and then burst fire again shortly later. So the neuronal signal, or the action potential, is transmitted as the generation and regeneration of these action potentials. So this signal that is going down the axon is essentially a generation and regeneration of an action potential traveling down, which is just a massive depolarization. So a good example of action potentials in signal propagation is puncturing the skin. Nerves can be stretched, allowing for stretch-sensitive ion channels to open, initiating and propagating an action potential. Then that information is communicated to the next neuron across the membrane. So one emerging picture that you should be getting at this point is that the brain is an interconnected mesh of membranes, where an action potential in one is an electrical signal, which will then be changed and converted to a chemical signal, which can then talk or communicate with a postsynaptic membrane or a postsynaptic cell. So after exam 1, what we're going to start to do is explore what happens when the action potential arrives at the presynaptic terminal. So we've talked about the action potential and how it travels down the neuron, but what actually happens when it gets to this point, when it gets to the end of the neuron? And what happens when we have a connection to another cell? So here's like a dendrite of another cell. Here is the cell body right here with other dendrites going to other areas. And then this postsynaptic cell has an axon of its own and will go and make connections to other cells. So what happens when the action potential arrives at this end? Well, in chapters 5 and 6, we're going to discuss the release of neurotransmitters. There's a lot of neurotransmitters and the various systems that exist within the brain. So these first two exams and these first two sections of this course are going to be very, very important that you understand. Mainly due to the fact that action potentials are what are going to lead to the process of exocytosis, which leads to neurotransmitters being released. And all the interactions that occur within the brain due to drugs at the level of the synapse are going to be crucial in understanding different neurotransmitter systems and why certain drugs do what they do. And this is going to carry on even into 376, which is the second part of this course, where you're going to be expected to know what these neurotransmitters do and what they are responsible for. So I will have the first exam up and ready to go after this lecture, hopefully, and that is due in 2 weeks. You'll have the weekend to do it. Again, make sure that it is done on time, as I do not allow for exams to be made up because you get an entire week to do it. If you have any questions about the exam, I can't answer direct questions, but I can try to clarify some points. And I hope that I wish you best of luck on that, and then we will continue on to our discussions on neurotransmitter systems.